Welcome to AI Explains.
Today we're doing a deep dive into a paper from 2019 by Hafemeister and Citation.
Yeah, and this one really, it really changed the game for anyone working with single-cell data. It's called Normalization and Variant Stabilization of Single-Cell RNA-seq data.
It sounds super technical, and it is, but what it solves was this really fundamental problem.
Which is what, exactly?
How to tell the difference between what a cell is actually doing, its biology, and just the noise from the sequencing machine itself.
Right, and it all comes down to this idea of sequencing depth. Let's try to explain this simply.
Sure.
Imagine you're a baker, and you want to figure out a recipe. You got two things in front of you. One tiny little chocolate chip cookie and a giant sheet cake. And your job is to just count the chocolate chips. Well, the cake is going to win every single time, you know.
Of course, it might have 1,000 chips to the cookie's 5. But that doesn't mean the cake's recipe is more chocolatey.
Exactly. It just means the sample is bigger. In biology, the cake is just a cell where the machine happened to read a lot more data.
And for a long time, the fix for this seemed obvious. It was called log normalization.
It's basically take the number of chips and just divide by the weight of the cake. Problem solved, right?
You would think so.
Sure.
But what this paper proved is that this simple division, it actually messes things up in its own way. It assumes if you double the size, you double the counts.
And biology isn't that clean.
Not at all. The old way was actually distorting the data. You were still seeing patterns based on the size of the sample, not the real recipe.
Okay, so that brings us to the big solution in this paper, a tool called SCTransform.
Right. Transform just. It stops trying to divide the problem away. It does
Sure.
something much smarter. It uses a method called regularized negative binomial regression.
Whoa, okay, let's unpack that one. Regression means it's building a model, a
Yeah.
prediction.
Precisely. For every single gene, the computer builds a tiny model. It asks a simple question. Given how big this cell is, how deep we sequenced it, how many copies of this specific gene should I expect to see?
So it's a personalized prediction for each gene, but what's the regularized part? That sounds important.
It's maybe the most important part. Single-cell data is famously noisy and sparse.
A lot of empty values.
A ton. So if you try to build a model for one gene using only that gene's data, you might just be modeling noise. You'll overfit.
So you can't trust it.
You can't. Regularization fixes this by pooling information. It looks at a bunch of genes that have a similar average level and kind of borrows strength from them to make one smooth, reliable prediction line.
Ah, so it uses the wisdom of the crowd to steady the individual. So once you have that prediction, what's next? How do you find the biology?
You look for the surprise. The tool calculates something called the Pearson residual.
The residual is the difference between the prediction and the reality.
Exactly, the surprise score. If the model predicts you'll see 10 molecules, and you actually see 10, the residual is 0. That's boring. That's just technical noise.
But if the model predicts 10 and you see, say, 50...
Well, that's interesting. That high score, that high residual, isn't from the machine. That's your biological signal. That cell is special.
The paper gives some great examples. There's a gene, MEL-AT1.
Yeah, MEL-AT1 is everywhere, in almost every cell. The old method made it look like it was bouncing all over the place.
Yeah.
Transform showed that, no, that was all just technical noise. It correctly flattened it out.
And the opposite example was a gene called PPBP.
Oh, this was the smoking gun.
Yeah.
PPBP is a marker for a cell type called megakaryocytes. The old method was so confused, it actually created a negative relationship.
Meaning?
Meaning it suggested that the more data you collected from a cell, the fewer PPBP molecules it had.
Which is?
It's physically impossible.
Yeah.
Transform immediately fixed it.
And you can see this so clearly in the final pictures, the UMEs.
Absolutely. Before, the maps of the cells just showed clumps based on sequencing depth. All the big cells hung out together.
A map of a technical error.
Right. But with Transform, suddenly the cells clustered by their true type. T cells found other T cells, B cells found B cells. The real biology just popped out.
It's incredible. It finally separates how well the machine worked from what the cell is actually doing.
And it's now standard. If you use the Seurat toolkit, this is built right in. It's the gold standard.
It does make you wonder, though.
What we might have missed.
Exactly. If our math was basically broken for years, if it was hiding real biological signals behind a wall of technical noise, what have we overlooked?
What rare cell types or subtle signs of disease were just washed out by a bad calculation?
And now that we have the right lens, what are we finally going to see?